Dear students, welcome to online class of biology. So, dear students, we were studying about various topics like the DNA, its replication, transcription and translation, and we have also studied about the control of gene expression, genetic code, etc. And now we are going to study about a new topic, that is Human Genome Project. So, Human Genome Project means what? It's a project only, okay, to understand the whole human genome. In a cell there are 23 pairs of chromosomes in each cell of human being. So to know the entire content of the human genome, okay, that or to understand the sequences of bases, okay, or genetic makeup of the human being, that was the major aim of the human genome project. Okay, so if there are two individuals, they are different from each other, and their DNAs also will be different, isn't it? At least in some points. Actually, after the human genome project, it was found that 99.9% of DNA of two different individuals of the same species, they are similar, but only a few part they differ. Understood? So this human genome project was possible due to certain techniques like genetic engineering. Understood? Then what it was means due to genetic engineering advancement, okay, or development of genetic engineering had helped the scientists to isolate and clone any piece of DNA, and that helped, okay, in the process of human means in this project, okay, human genome project. Actually, it is a very large project. Just imagine, that is, a human being consists of, or human genome consists of 3 into 10 to the power 9 base pairs, we have learned in a haploid set, isn't it? And the cost of this will be very high to understand the whole set of genome, isn't it? It is very costly, and it is estimated that, okay, around 9 billion US dollars, that much it may come. And one more thing, if we the obtain the sequences, okay, if we are storing that in the form of a book, okay, and if each page of the book contain 1000 letters, and each book contain 1000 pages, then 3300 such books are required to write the information. Just imagine, okay, this is simply some what facts means, the to make you understand what the importance of human genome project, okay. And also it says that what how much difficult and how much huge project it was, okay. So the amount of data, okay, after the analysis of human genome project, it is not easy to store in any normal way in copies or books. So a new biological branch also has evolved, that is bioinformatics, okay. So bioinformatics help to store the data, retrieve the data, and analyze whenever it is needed, okay. Then data which we get from the what analysis of human genome, okay, we can store in means with the help of high speed computational devices, okay. And also so that we can store, we can analyze, we can retrieve whenever it is needed. So this led to a new branch of biology that is known as bioinformatics. So let's see what were the goals of HGP, okay. HGP's goals were, first, to identify around 20,000 to 25,000 genes, okay. So genes, we have studied that part of DNA, isn't it, segment of DNA. So in human genome, it is identified that 20,000 to 25,000 genes only are there, and to identify them, okay, and then to determine the sequence of 3 billion base pairs, okay, 3 billion base pairs are there. We have already studied, isn't it? So this 3 billion pairs sequence, okay, from first DNA to 23rd DNA, that is actually first to 22 autosomes and XY chromosome, all this has to be analyzed, okay. So and its base sequences, okay, that is A, T, C, likewise sequences will be there, no? So how, what is the sequence of this nucleotides? Only 4 are there, 4 nitrogen bases are there, but how they are arranged in each what part of the DNA, like I told already, 26 alphabets are there, and with that, with the different combination, a number of letters can be produced, isn't it? So in the same way, in which sequence, in which combination they are found in the DNA, then store this information in databases, then improve tools for data analysis, transfer related technologies to other sectors like industries, means this can, okay, if we know the genome, the products can be understood. So if a person is suffering from some enzyme deficiency diseases, which gene is responsible for that, for the production of that particular enzyme, if we know, what can we do? We can make that, isn't it, in artificial medium. Just an example is what? Insulin. Now what we are getting is actually means humulin, understood? So when we know the gene sequence, we can what use, we are having a lot of use for that, understood? So that can be used in industries, okay, in health sectors, etc. Then another important issue was to address the ethical, legal and social issues, because when we know the genome, lot of problems can be created, isn't it? Any kind of unethical experiments can be done, isn't it? Cloning, if it is done, it will have lot of what in human being, it is if it is done, what will happen? It can create a number of problems, isn't it? If anyone is creating a a notorious criminal's clone, understood? So it will be difficult for the society, isn't it? So like this, to address the ethical issues, all this were the aims of the aim of the HGP, Human Genome Project, okay. So you have to learn this. Sometimes it can ask for 3 marks, okay. What were the goals of HGP? And it was a 13-year project and coordinated, okay, it was coordinated by US Department of Energy and the National Institute of Health, okay. US Department of Energy and the National Institute of Health, understood? And Wellcome Trust, another UK company, they also will be they also become a part of this, and other help, okay, or contributions came from other countries also, like Japan, France, Germany, China, etc., okay. And this project was completed in 2003, understood? And this led to a revolution in the way to diagnose, treat and to prevent the disorders, genetic disorders. You would have, we have studied, isn't it, to correct this. So like this, lot of lot of results was coming from this. So in addition to this, in Human Genome Project, non-human organisms, okay, that were also sequenced, their DNAs also were sequenced, okay, like what? Yeast, bacteria, Caenorhabditis elegans, that is actually a nematode, okay, Drosophila, plants like rice and Arabidopsis, etc., also were sequenced as a part of this, understood? So these were the major means events that took place in were in Human Genome Project, okay. So who coordinated this? US Department of Energy and the National Institute of Health, understood? And who had given the major contribution? Wellcome Trust, okay. And then other contributions came from different countries. So when we talk about the methodologies, okay, there were two different methods or two major approaches, okay, to do this. They are expressed sequence tags or ESTS, okay, and other is sequence annotation, okay. So expressed sequence tags means what? Here only the means focus was on the genes that are expressed as the RNA. We have learned now about the exons and introns. So whichever the DNA sequences are expressed or genes are expressed, they were only focused. Understood? So the other one was simply sequencing the whole set of genome that contained all the coding and non-coding sequence. Okay, understood? So these two approaches were there. First is what? Expressed sequence tags, where only the expressed genes were focused, means the identification of the expressed genes only were the aim of that approach. Okay, and the other one is what? Sequence annotation, where expressing, non-expressing or coding or non-coding sequences were also sequenced. Understood? For sequencing, what they have done is, first, a long molecule cannot be sequenced, or it is, as DNA is a very long molecule, and it is very difficult to analyze the sequences of them in its all length. So what approach they have done? First, they have cut the DNA into smaller pieces, okay, so that it can be cloned into host. Okay, so first, what? DNA, human DNA got cut, means after isolation and all, okay, after isolation of DNA from the cell, it will be cut into small fragments, and then it is cloned into suitable host using vectors. Okay, so what is the importance of this? That is, you can get a number of copies when these human gene fragments or DNA fragments, when it is cloned into host using vectors. Okay, when this host, like what yeast, bacteria, etc., they multiply, you will get a number of copies of the same DNA or gene, isn't it? So you can do that experiment again and again to sequence that particular fragment. Understood? So this was the approach. So the commonly used vectors were bacteria and yeast, and the vectors were bacterial artificial chromosomes and yeast artificial chromosomes. Okay, BAC and YAC. Okay, they were the vectors. What for it is used? To to give the DNA fragment of human being into the host, like bacteria and yeast. Okay, so bacteria and yeast were the host. Into that, what is entered or what is attached? Human DNA. Understood? Fragment of human DNA, not the full this one. Fragments of DNA were attached. Understood? Using what? Vectors. Vectors like plasmodium, malarial disease. Plasmodium is transferred from infected person to healthy person by what? Mosquitoes. Mosquitoes are the vectors there. So just like this, the human genome fragments were taken by this BAC and YAC. Okay, and it is given into the yeast and bacterium host cells. Is it clear? Then the fragments were sequenced using automated DNA sequencers, okay, that were developed by Frederick Sanger, and it was working on the principle of overlapping. Okay, method of overlapping. Understood? So this was the approach, or this is the way how they have done. Understood? Then specialized computer based programs were developed, okay, so that these sequences were the data that was getting from the sequencing can be stored. Okay, and then these sequences were subsequently annotated and assigned to each chromosome. Understood? So overlapping fragments, okay, were found out, and on that basis, understood, they were sequenced, and the data was stored in the computer. Understood? And the sequencing of chromosome 1 was completed in 2006. Okay, and it was the last chromosome to be sequenced, and it is the biggest chromosome. Okay, then all the 24 chromosomes, that is 22 autosomes and XY. Okay, these all were completed, and this project is known as what? The Human Genome Project. Okay, so and the genetic and physical map of the genome also was done. Okay, so for this, the information of the polymorphism, okay, of restriction endonucleases, recognition sites, okay, repetitive DNA, all this we will study in the next topic, that is in the DNA fingerprinting. Understood? So polymorphism of restriction endonucleases, recognition sites, okay, repetitive DNA sequences called microsatellites, etc., were useful in making the genetic and physical map of the genome. Understood? So after completing the genome mapping, okay, the conclusions or observation done were human genome contains 3,164.7 million base pairs. This much base pairs are there. The average gene means there are big genes, there are small genes. So an average gene consists of 3,000 bases. Okay, and the largest gene is found to be dystrophin with 2.4 million bases. Okay, then the total number of genes was estimated as 30,000, and it was just very less than the assumption that it contains 80,000 to 1,40,000 genes. Means it was the assumption that human genome may contain 80,000 to 1,40,000 genes, but it was found that only around 30,000 genes are there. Okay, and another important feature that was found is 99.9% of the nucleotide bases are exactly same in all the human being. Understood? Around 0.1 percentage only is different. Understood? So in a species of human being, all the members were having 99.9 percentage nucleotides same. Okay, nucleotide bases are same. Then the functions are unknown for over 50 percentage of discovered genes. Till now, sometimes news you can listen. The function of this particular gene is found out. Understood? So there are many more genes are there whose function is not known. Understood? Less than 2% of the genes codes for protein. You understand this point? Less than 2% of the gene is only necessary for coding the proteins, and the other ones are junk genes without any function. That forms the what introns we learned, isn't it? Then repeated sequences may make up very large portion of human genome. Repeated portion I told just above I told for genome mapping and all, it was very useful. So repeating genome means what? No, a particular segment of DNA with the same nucleotides. Okay, for example, ATGGC. Understood? So like this, it may repeat many times. Okay, sometimes 50 times, sometimes more than that, 100 times or 1,000 times. Understood? So repetitive genes are there. Understood? So this forms a large portion of the human genome, these, okay, I told 100 to 1,000 times it can be, and they are thought to have no direct functions, but they helped in the in the process of evolution. They were having what they have contributed, means the scope for evolution is still kept in our body, isn't it? So if this changes happens, there is lot of mutation can take place and can change our character only, isn't it? Then chromosome 1, that is the biggest means chromosome, and it is having 2,968 genes. Okay, and Y is having the fewest genes, chromosome Y, that is found of the males, and it is having only 231 genes. Okay, so and scientists, they were also able to identify around 1.4 million locations where single base DNA differences, that are known as single nucleotide polymorphism, means what only in one what nucleotide there will be difference. Okay, so these all were very revolutionary findings, and this could help, okay, in different ways in the identification of genetic disorders, in the treatment, okay, in the product, in the formation of various products of genes to treat the various diseases. Likewise, lot of what then DNA fingerprinting is based on this, okay, repetitive sequences. So many, it was a revolutionary finding. Okay, human genome project, when it was completed, okay, it was a very what huge step in the genetics. Okay, so this is about the human genome project. I think you have understood this well. Thank you very much.